DNA, T-cell, heart. Today I'm breaking down a new mechanistic model for rare myocarditis after COVID-19 mRNA vaccination, published in Circulation, October 2025. First, context. Myocarditis after mRNA vaccination is very rare. The benefits of vaccination still clearly outweigh the risks. This study doesn't overturn that. It explains how a rare event can happen. Here's the core idea. The vaccine trains T-cells to recognize the spike protein. For most people, that's the end of the story. But in a small subset, some of those T-cells also recognize a look-alike piece on a human protein in heart muscle, specifically a peptide from a cardiomyocyte potassium channel. That's molecular mimicry. One key fits two locks. When those cross-reactive T-cells activate, they can mistake heart cells for the virus. That's the autoimmune-like spark behind rare myocarditis. The team didn't stop at correlation. In mice, immunizing with the mimic peptide alone reproduced AMP-like cardiac inflammation. That moves the evidence from association to a causal, mechanism-supported model. Patient data mattered too. T-cells from people with vaccine-associated myocarditis showed an expanded cytokine profile, more like classic autoimmune myocarditis, compared to people who had COVID-19 without vaccine-related myocarditis. Now the twist: it's not only what T-cells recognize, it's where they go. The study found a subset of cMET-expressing T-cells that preferentially home to the heart. Block the cMET pathway in the model, and myocarditis doesn't take hold. Translation: antigen recognition plus tissue-specific homing equals organ-specific inflammation. Remove the homing, and the fire fizzles. Why this matters if you build vaccines or immunotherapies. Mechanistic clarity, mimicry, TCR specificity trafficking. Safety insight, biodistribution antigen expression pattern. Translational relevance, oncolytic viruses, cytokine drugs engineered T-cells. Potential intervention, cMET emerges as a target to prevent heart-tropic T-cell infiltration. Actionable takeaways for the biotech toolbox. Screen early for epitope mimicry with human proteins. Track T-cell homing receptors, like cMET, in preclinical studies. Use vectors and promoters that limit unintended tissue expression. Treat biodistribution as a primary safety variable, not an afterthought. Balanced view. Infection risk remains higher than vaccine risk. This work refines the map, not the destination. Vaccines remain a cornerstone. Summary: spike-induced T-cells can cross-react with heart antigens. cMET-positive T-cells home to the myocardium. A mimic peptide induces AMP in mice. Blocking cMET prevents disease. Read the study at aheyjournals.org.